Um, it's a pleasure to be here. Thanks, Matt, for inviting me. So this compound is bromodichloromethane. It's part of the trihalomethanes. It's a disinfection byproduct of drinking water. Now, what the audience might feel is drinking water disinfection byproduct. You will remember that maybe in the 1980s or the 1990s, NTP was probably done with the kind of toxicity studies that was there in disinfection byproducts of drinking water. Why this guy somehow is raking the whole thing up again? There was no need for it. Well, there were some reasons. Disinfection byproduct of drinking water, especially bromodichloromethane, dibromochloromethane, bromoform, these were linked to hepatotoxicity long back. But they were at very decent doses, very high doses, in fact. And it was hepatotoxic to normal, probably hepatotoxic to normal healthy individuals. But in around 2009, 2010, I had an idea that maybe these hepatotoxins can be of interest if they are provided, or rather exposed in low doses in underlying condition of obesity, because as an immunologist, I can actually go into stating that obesity is a low systemic inflammatory trigger. So if that is the case, then probably these can act as a second hit. So luckily for me, my Pathway to Independence Award was funded, and I don't know who was the person that reviewed it, made my day, and finally I am here after 3-4 years, publishing a decent number of papers on bromodichloromethane and its effect on development of non-alcoholic steatohepatitis. So CYP2E1 is a major P450 isoform involved in the bioactivation of BDCM to reactive intermediates. This was proven long back, and in fact my postdoctoral mentor Ron Mason, when he saw this, he said, well, you give this to me because I worked on CCL4. I said, Ron was kidding. Ron is a... So he said that, yes, this probably has a future for you. So the dermal contact of this particular compound can also lead to greater systemic BDCM doses than water ingestion. A lot of studies have been published with bromodichloromethane and systemic exposure, especially from swimming pools and daily showers. And that has been found to cause some reproductive birth defects, epidemiologic studies which were recorded long back. But then, I established my lab and worked straight with bromodichloromethane and how it actually can produce a second hit in an underlying condition of obesity, which we use a high-fat diet, 60 kilocalorie %, which is quite high. Not a typical Western diet, but the typical diet-induced obese model, custom-made, which is marketed by Jackson Laboratories. And in a series of publications, we are still publishing bromodichloromethane. We have actually carved out a mechanism for bromodichloromethane toxicity and bromodichloromethane as a second hit for non-alcoholic steatohepatitis progression, including the earlier sinusoidal endothelial dysfunction, the inflammatory phase, and the fibrogenesis phase. But then, a big question. What I was trying to do, is it a self-fulfilling prophecy? Does bromodichloromethane really does something? Now, most of the people in the audience might be thinking, okay, rodent models produce, no other person in the whole world is working with bromodichloromethane, and only the results coming out are from my lab. So is it a self-fulfilling prophecy? Finally, this year, there was a good study with Harvard School of Public Health, in the Cypress program of the Harvard School of Public Health, published a detailed cross-sectional study and an exploratory analysis where they found that brominated trihalomethane, specifically bromodichloromethane, had a direct relationship with, or rather a causative link between type 2 diabetes mellitus and other probably NASH factors. So that, and let me tell you this, that when I talked to Konstantinos, who is the main person behind this study, he said that probably your studies which were produced in the lab were instrumental in setting up this analysis, so which was kind of a boost for my laboratory and my ideas. Now, looking into this, then we tried to dig into the enhanced data and look from 1999 to 2006, and where we have also found that in non-drinkers, which were, and of course, which is a very important factor when we are looking into NASH progression, an adjusted odds ratio of 4.1 is prevalent, and which actually links with the higher serum ALT values. Though ALT values may not be a good predictor, sometimes in NASH, as the speakers in the beginning sessions have spoken, but still ALT values are taken as a clinical indicator of NASH, and of course, progressive NAFLD. So this particular study is going out, and we actually can say for sure that epidemiology is also pointing to the fact that brominated trihalomethanes are a risk factor for the development of the disease. Presently, now because those are all published data, presently we are focusing on two important things. And the big question that we are asking is whether BDCM-induced oxidative stress causes experimental NASH by targeting the epigenome. Now this is a big, big question. Nobody knows what we will end up in. But for the working hypothesis, one thing is that whether BDCM induces CpG island methylation of gene promoters. The second is whether there are microRNA inductions, specifically microRNAs which are involved or have a pronounced role in inflammation. That is microRNA 21, 155, and 34A. So the approach is that, as most of us do, to hand over to a particular company who does a CpG island array. We did not do that in the lab. And then also we seconded that with an MIR array. So the results that we looked into about the CpG island array was there were 26 genes that were methylated in the promoter regions. 2 genes, and out of those 26, which had a good confidence level, 2 genes were identified in our lab to be correlating with the data that we are generating. And out of that 2 genes, one was TRPV4. We didn't understand why TRPV4, which is a cation channel protein, has anything to do with NASH indicators or BDCM toxicity. And the other one was DOC2B. DOC2B, as you know, is very much related to insulin uptake and insulin role in glucose uptake. So what is TRPV4? TRPV4 actually, when you look into the existing literature, is a transmembrane cation channel proteins, mainly responsible for influx and efflux of calcium and sometimes magnesium. But it is a very important transmembrane protein in sensing the changes in osmolarity. The second one is it changes. It actually senses membrane alterations and also is responsible in preserving the membrane integrity. So having said that, we looked into the TRPV4 methylation patterns, whether we can reproduce what we have found out in the array. And as you look in the data here, now this was carried out in the lab in generating the methylated specific PCRs. And here what we found is that in DIO plus BDCM in 1 week, we have this expression of the methylation here. And also this was further increased in DIO plus BDCM, which was at 4 weeks. CYP2E1 knockout mice had a lesser effect on the methylation patterns here. The next, what we looked into, whether the specific cells have anything to do with the TRPV4 mRNA expressions, because methylation really doesn't mean anything if there is no significant changes in the mRNA or the protein expression. So what we looked at is the cell-specific expression here, and hepatocytes did the mRNA expression of TRPV4 really went up, and not so much in the Kupffer cells, but definitely remarkably high in stellate cells. Then we analyzed the whole liver homogenates for the expression of TRPV4, and here you can see that in DIO BDCM groups, actually there is maybe around 1 to 2-fold protein expression, a higher protein expression, whereas in CYP2E1 knockout, it's not there. So at this point, we were wondering whether the methylation in the TRPV promoter methylation increase in mRNA and protein levels does they have any significance whatsoever. However, most of the methylation data that has come out, I mean, we were shocked by this. Methylation is responsible for silencing genes. Most of the times. I was going around the country trying to find out a person. I'm not an epigenetics person. My postdoc was working on it. So I personally went out and reached out to people and said, hey, look, what is this happening? We are getting a higher expression. Is the methylation has something to do with it? Then there were a number of papers which were shown to me by experts, and they said that there are ways to do it. And it may be that there are certain binding proteins, like CTCF, which can actually bind to the CPG islands. Normally, this is the case. However, in order to prevent the enhancer from going on and triggering the expression to go forward. However, if you have a methylation in the CTCF binding region, then probably this is stalled. So we looked at and through a software analysis, we went and tried to get a sequence matching our array, whether that particular sequence matched the CTCF binding CPG islands. We did find, but it is still at a very experimental stage. We have not confirmed that in big details. However, what evolved from these experiments is the molecule TRPV4. So because we thought that this protein is going up, it might have something to do with the BDCM toxicity. So what we should do, we went ahead and asked two definitive questions. Why TRPV4 may be methylated in the promoter region following BDCM exposure? And what might be the significance of its upregulation in the liver? So we did two simple experiments. We tried to see the expression in both the Kupffer cells and the stellate cells. We found that in Kupffer cells, this particular TRPV4 expressions are there when you have BDCM exposure. In stellate cells also, as you can see, these are, the green actually is alpha smooth muscle actin. These are activated stellate cells. And then the red is the TRPV4, and you can actually see that they perfectly colocalize, signifying that the TRPV4 are expressed in these two kinds of cells. TRPV4 also has been seen by our lab to be expressed in hepatocytes. And they have a very interesting role in hepatocytes, which we are not describing here today because of the shortage of time. They might play a role in the fat deposition, but that is something different. So in order to find out the role of the TRPV4, we thought to use TRPV4 knockout mice, TRPV4 antagonists. TRPV4 agonists, I looked at the literature. There was absolutely no way we can use TRPV4 whole body agonists because they are reported for toxicity and pulmonary edema. So we were left with only two ways of trying to find out what TRPV4 might do. So first approach was TRPV4 knockout mice. So went on hunting again for the TRPV4 knockout mice. So naturally, in Duke neurology, Wolfgang Lüdke is the person who actually made these mice. And he was very happy to give those mice colonies to us. And then the thing started. So remember, at this point, we were thinking that TRPV4 is playing a role in BDCM toxicity. And these are the type of mice that we use. I mean, they are raised in BL6 background. And so these are the lean-fed mice. These are the DIO 60% kilocalorie, and these two, and one are which are exposed with BDCM. So everything the mice arrived in the lab, and we tried to look into the effects. We were shocked by the results. TRPV4, DIO plus BDCM, I mean, we have this phenotype where we see some necrosis as we go forward. With those particular dose that we are using, we actually see that there is hepatocellular necrosis, et cetera, et cetera. But look at the TRPV4 knockout mice. These mice in 1 week developed a type of hepatocellular necrosis which we do not find even at 4 weeks. So our hypothesis crashed. TRPV4 is not aiding in the toxicity of BDCM. Rather, it is doing a job probably which means that it has a protective role. We looked at serum ALT. We looked at HMGB1, which is a DAM. Here, everywhere, the TRPV4 knockout mice at 1 week was showing a response which we have never estimated, which we have never looked into. And so naturally, it opened a wide area of research, intense research, more interesting and enthusiastic research into this particular area. Then we looked at whether it was doing anything for the lipid peroxidation that usually happens. Here also you can see the lipid peroxidation actually, I'm not showing the morphometry here, but if you look at it is actually a 5-fold increase than the DIO BDCM at 1 week. It is almost correlated with the DIO BDCM at 4 weeks, but still it is more than the DIO BDCM at 4 weeks. When we look at the 4-HNE histidine adducts, which is a stable form of the adducts, we found that there is a significant increase in the 4-hydroxynonenal histidine adducts in TRPV4 knockout at 1 week as compared to DIO BDCM at 1 week. Next, we went to find out whether there was any change in the inflammatory things. So we looked into IL-1 beta, and there IL-1 beta was significantly increased in TRPV4 knockout mice at 1 week, and almost this is like 6-fold. The mRNA expression of IL-1 beta and TLR4 are huge. TLR4 is around 25-fold as compared to, almost like 20-fold compared to DIO BDCM at 1 week. And even it actually is greater when we compare that with 4 weeks. Now, IL-1 beta secretion actually tapers down as NASH progression goes on. This happened in both cases of TLR4 and IL-1 beta. The next we looked into was we isolated the non-parenchymal cells. We did not separate them further into Kupffer cells and other cells, but we just took the non-parenchymal cells. And here we did a FACS analysis of the MHC class II expression and the CD86, which are both very important mediators for the adaptive immune response. Now there, what we see is that there is almost like a 28.6%, is almost close to 30% increase in the MHC class II expression, sorry, the CD86 expression, and also there is almost a double increase in the MHC class II fluorescence, which actually indicates or suggests that TRPV4, absence of TRPV4 rather, plays a significant role in increasing the immunological responses that are taking place due to BDCM exposure in a condition of obesity. Kupffer cell activation, as we all know, is a very important player in NASH pathogenesis. And there, we actually looked into CD68, which is a marker of activated Kupffer cells. We also looked at F480, but I'm not presenting the data here. The CD68 shows a 3-fold increase in the TRPV4 knockout mice, while there is, as compared to DIO BDCM at 1 week. Another important factor, MCP1, which is also recognized as a vital mediator for NASH pathogenesis, also went up very significantly higher, though we did not see that kind of a drastic mRNA effect in the protein. In protein, it was maximally at around 7 to 8% increase, a 7 to 8-fold increase. Stellate cell activation is a very important part of the NASH pathogenesis. We all know that. I'm not going into the details of it. We looked at alpha smooth muscle actin, which is the activated stellate cell marker. Here, there is definitely an increase, as you can show in the immunohistochemistry here. TRPV4 knockout mice at 1 week, with BDCM at 1 week have significantly higher alpha smooth muscle actin expression than TRPV4 the wild type mice at 1 week, suggesting that all the indicators of NASH progression have a very increased, a significantly increased phenotype in TRPV4 knockout mice. So at this point of time, we were actually clueless. Okay, fine, we have got this beautiful data. TRPV4 knockout mice have very high increases in these, and there is an augmentation of NASH pathogenesis. But why is it doing so? Or how is it doing so? Is TRPV4 expression a mere adaptive response, which can happen to many number of proteins? We don't know even so many number of proteins. Or is it targeting the oxidative stress, which is through CYP2E1, after it is induced by the same? Sometimes these things are happening. These are very normal phenomena, that one protein, which actually is upregulated because of an adaptive response, but whether that has a particular role in turning that loop off. So we were looking into, we started looking into whether it has something to do with the CYP2E1 protein and its activity. So there we used a simple experiment again. So we've used DIO mice, which were TRPV4 knockouts. We exposed them to BDCM, but at the same time, we used an inhibitor of CYP2E1, which is diallyl sulfide. And we exposed these mice, treated these mice with DAS as well as BDCM at the same time. And what we found was we could significantly change the way the mice behaved. As you can see in this image, you don't probably need any indicators to say that these mice which are sick, you can see at 1 week, TRPV4 knockout mice. Here, these mice are up and running. So this was a very significant result. And as you can see from the indicators, the same mice at 1 week which are treated with diallyl sulfide, the IL-1 beta response is down. We looked also at TLR4. We looked at CYP2E1 mRNA expressions. Those CYP2E1 mRNA expressions here really doesn't mean anything, but definitely there was a decrease, a significant decrease in the CYP2E1 induction, the mRNA induction. Next, we tried to assess the CD68, which is the Kupffer cell activation. And here, it is very clearly, you can see that there is a decrease in CD68 after you have diallyl sulfide injected mice. MCP1 was also significantly decreased. The same thing happened with alpha smooth muscle actin. As you can see here, diallyl sulfide actually significantly brought down the alpha smooth muscle actin levels on those livers. And finally, we also looked at the basic pathology here, and you can actually see that the TRPV4 mice, which are injected with diallyl sulfide, do not have the infiltrative phenotype that we see with TRPV4 knockout mice exposed to BDCM at 1 week. 4-hydroxynonenal, the oxidative stress, which is a direct consequence of CYP2E1 activity. We did not, just to mention here, we did not measure CYP2E1 activity because we did not find very specific assay to measure CYP2E1 activity, which will only inhibit CYP2E1 and not the other enzymes or drug metabolizing enzymes. So here, 4-hydroxynonenal was significantly decreased. So was the serum ALT levels, which was very decreased, and also the 4-HNE histidine adducts. So at this point of time, we summarized our data in terms that TRPV4 mRNA and protein is increased following BDCM exposure, which was quite evident. Absence of CYP2E1 decreases TRPV4 protein expression, which happened, which actually I'm not showing the data here. TRPV4 is expressed highly in stellate cells and Kupffer cells following BDCM exposure. Absence of TRPV4 greatly enhanced hepatic inflammation, early fibrotic phenotype, and blocking CYP2E1 in TRPV4 knockout mice attenuated enhanced inflammation and early fibrotic phenotype. We have data with the fibrogenesis, but I mean, this is probably too short a time to describe all those things. So now, with this summary again, it really doesn't mean anything. CYP2E1 it is doing something. So it might be that CYP2E1 mediated oxidative stress increases TRPV4 protein as an adaptive and protective response against the cellular damage when we are exposing to BDCM. Increased TRPV4 blocks the induction and possible activity of CYP2E1 through a feedback mechanism. That's what my hypothesis is, probably by an unknown mechanism. Now, here we should recollect that TRPV4 is a calcium channel. So probably calcium signaling is getting activated here because of the higher expression of TRPV4 in these exposed mice. And this calcium influx can actually do a lot of things in order to try to mask the CYP2E1 effect or restrict the metabolism of the BDCM, which is there. Now, interestingly, we have reproduced the results in both acetaminophen and alcohol-induced liver injury. So it is not specific for a particular toxin. So it has to do something with CYP2E1. And TRPV4 might be a master sensor of hepatotoxicity by regulating CYP450 enzymes. We have not checked the other enzymes, but it might be a possibility. The mechanism, we have worked out that, but probably not today. I will not explain it today. So I would like to acknowledge the funding. So I am funded by the R00, the NIH P20, where it is just funded, and the Aspire 1, which is part of the University of South Carolina, the CAM Center at the University of South Carolina, and of course the NHS departmental funds. Like to acknowledge the collaborators, Dr. Deal, who has been supporting me for a number of years now, Dr. Litke, who has been very supportive of the work, Jose have been very supportive, and Dr. Darwin Bell, who gave us the mice from Medical University of South Carolina. This is the group. Actually, this work is being carried out by my postdoc, Dr. Seth, and also one graduate student, Dr. Deepto. And I'm ready for questions. Yes?
What dose are you using?
So here we have two models. One is in order to induce systemic injury, we inject around 80 or 100 parts per billion to these mice, which is the EPA permissible dose. The other one is we give them through drinking water by oral gavage, because we are specifically looking into the drinking water responses. Now, we have a separate project with BDCM where we are looking into the TLR4 gut TLR4 axis. So there we actually expose them to through drinking water. Yes. Okay.
So data are really interesting. The vilanoid receptors, there's several of them, and we've looked at TRPV1, which also regulates calcium flux, and see just the opposite results. So the vilanoid receptors usually activate neuroinflammation.
Mm-hmm. Pain.
Right.
Yeah.
And so if you used any kind, instead of using knockout mice, if you block calcium channel signaling, for example.
Yeah, that we have done. We have blocked we were using calcium channel blockers, simple calcium channel blockers. We did not get anything. We did not get the results here. But when we used TRPV4 antagonists in higher doses, which were like knocking the TRPV4 down, there we got the type of responses that we have. So So it has to do specifically with TRPV4, probably through calcium. But what we have found out is that TRPV4 is actually activating enos phosphorylation. And we have recently published, in fact it is in revision, it's almost acceptable, that in fact that BDCM induces a defect in enos phosphorylation, and the nitric oxide that is there actually, which is not there, in fact has an unrestricted role in CYP2E1 metabolism of BDCM, because Saurahb and his colleagues are famous for looking at nitric oxide inhibiting CYP2E1. So we have injected these mice with nitric oxide donors, and we found protection, primarily through blocking of CYP2E1. Now we are also looking at the transporters, the ABCB1 transporters. And that is another way of looking at it, because we don't want to look at only CYP2E1 here. Because as I have shown in the data, this is not a BDCM specific response. This is a drug metabolizing enzyme mediated response. We have found that in alcohol. We have found that in acetaminophen. And so what is big in this particular project is that TRPV4 probably is an adaptive response, but it has a role to keep the metabolizing enzymes in order the toxicity check. So far, we always know that the drug metabolizing enzymes are the center stage, but TRPV4 can take the center stage much upstream of these drug metabolizing enzymes. Yes?
Beautiful work. I just have a question. You use the 2E1 pharmacological inhibitor, right? When you inhibit 2E1, you use a pharmacological inhibitor.
Yeah.
Can you actually see the same thing if you breed the 2E1 knockout mice to the...
Actually, CYP2E1 knockout mice, when we use, we don't see the toxicity of BDCM. So we cannot use a double knockout, because first of all, CYP2E1 knockout mice are actually, they are cryopreserved. Getting these mice will take 2 years, and getting that a double knockout for TRPV4.
So if 2E1 knockout mice is not sensitive to toxicity, but how can you conclude actually sort of the 2E1 is the effector enzyme?
No, we are not telling that CYP2E1 is primarily the one. We are trying to say that all these enzymes which are necessary for drug metabolism probably the TRPV4 might have an effect on all of them. So it's not a CYP2E1. See, this work, it is just because BDCM is targeting CYP2E1, we are looking into this effect. But I have an idea that it has a role to play on all the CYP450s. TRPV4 is not a very specific thing for CYP2E1. That's my take on it. And we are looking into specifically the most abundant drug metabolizing enzymes on that.
Okay, but one more question, Jose.
Just a quick one.
Yeah.
So, could you talk, we might have discussed this before, but can you talk about the potential NERV2 oxidative stress or threshold for oxidative stress in the TRPV4 mice? Have you looked at NERV2 basal expression, how robust responses in response to anything that produces oxidative stress in these knockout mice?
NERV2 goes hugely high in these mice. NERV2 goes high, probably, and that we have seen that. Like initially, whenever there is oxidative stress or an agent that induces oxidative stress, there is a higher response of NRF2. But that we are seeing that same in this also. There is an increase in P47 FOX expression, NADPH oxidase, but probably what I have an idea is that NADPH oxidase is downstream of the lipid peroxidation, not upstream.
Okay, well, thank you.
Thank you.
We can discuss